next speaker online, Dario Valenzano from recently moved to Jena. Are you there, Dario?
I am here. Can you hear me?
Hello, sir. Welcome to ARDD 2021.
Thank you very much.
We're very happy to have you here. Whenever you're ready, you can start sharing your screen. That's perfect. Thank you so much.
Excellent. Thank you so much. Thank you, Morten. Thank you, Daniela. Thank you, Alex, for the invitation. It's an honor to be here. Sorry I couldn't be present in person, but it's great to be part of this event. So today I will tell you a little bit of our recent work. We largely use African turquoise killifish as our main model system to address questions in biology of aging that may have some relevance also to this audience. So the reason why we use this short-lived vertebrate is twofold. They are experimentally accessible systems. We can raise them in large numbers in the laboratory. And they display a very vast array of spontaneous age-dependent transformations that resemble human aging. And I hope I will convince you that the way in which killifish age can help us understand the molecular basis in which we age. And since they have such a short lifespan, they are accessible to experimental interventions. And being vertebrates, just like us, experimental interventions that postpone the onset of the major aging phenotype or the delay the onset of this phenotype may have medical relevance for humans. Okay, so it's very exciting for us to see an organism age before our eyes. And this is the greatest, I think, advantage of killifish. So from different perspectives, even at systemic level, multi-organ level, killifish undergo aging. For example, if you look at bone skeletal phenotypes, killifish in a matter of a few weeks, couple of months, will undergo loss of bone mass. So this is work done together with Björn Busse in Hamburg. And what Björn's team did was CAT scans in fish skeletons. And they could measure density drop, bone density drop, sporadically occurring with aging. This is work currently in revision, and we were excited because killifish display, even though they don't experience the same gravitational forces as we do because of their aquatic medium, they still undergo dramatic spontaneous loss of skeletal mass. This is work done previously by a postdoc, phenomenal postdoc in the lab, Gaurav Ahuja, who has his own independent group now in Delhi, in India. And so what Gaurav did was to study heart aging in killifish, and he found that killifish from young to mid-age to aged undergo an accumulation of autofluorescent pigment known as lipofuscin. This occurs sporadically in the heart, and also heart get infiltrated probably with fibroblasts that undergo increased signal for senescent associated beta-galactosidase as indicated in the bottom panel. And this phenomena occur spontaneously in killifish. One of the most striking age-related changes that occur in killifish is the display of brain age-related changes. Killifish have this unique feature among vertebrate, together with humans probably, to undergo very dramatic neurodegeneration in a relatively short time. This is actually A-beta-42 staining in the brain and phospho-tau, which are hallmarks of Alzheimer's disease. Neurogenesis has been shown to decline with age in killifish, and a recent paper from a couple of years ago by a Japanese group led by Hidekai Matsui showed that actually killifish undergo spontaneous degeneration of dopaminergic and noradrenergic neurons, which are actually phenomena associated with the onset of Parkinson's disease in humans, and they could manipulate genetically these organisms as well. Just this month, there was a very interesting publication in Aging Cell by Lutke Artgen's group in Belgium, where they show that actually killifish brains, these are actually cross-section of the killifish brain, actually they do pretty bad when it comes to responses to injury as they get older, and all the cells that are normally required to respond to brain injury in the young actually are not able to proliferate, differentiate in a proper way in older killifish brain, and they respond undergoing senescence, indicated here by senescent associated beta-galactosidase, again, this time in brains. There is an increased signal, and these actually are the levels, the expression levels for P21 and P27 that are dramatically increased in older individuals compared to young. So overall, there is this very striking aging related transformation happening across killifish organs. Our own group actually has recently started to focus their attention, for reasons that I will tell you in a few slides, to the gut. And so these are cross sections of the gut. These are sagittal sections of the gut. And what we have observed, this is work done by 3 phenomenal students in the lab, Quinn Kesenberry, Miriam Popkis, and Jens Seidel. What they studied actually is that by staining guts with Picrocereus red, fast green, they found that killifish undergo a dramatic increase signal of collagen deposition. So here in pink, you have collagen, and in green, so you have fast green marked cells. And so you see that this pink signal dramatically increases with age. This is actually quantified. Old fish have more of this pink pigment indicating collagen, and also performing metabolomics in killifish gut sections. This is work done by Miriam in particular, show that there is actually an increase in 4-hydroxyproline, which is the major component of collagen in killifish intestines. So doing proteomics in killifish intestine between young and old also shows very striking overexpression of collagen 6, for instance, as well as FREM to be another very important component, gene involved in the composition of extracellular matrix, as well as important chaperones. But Go analysis, in particular cellular components analysis, really shows as the most dramatic term there in between young and old fish, increased deposition of collagen. So is this aging, or is this compensatory? Is this dysfunctional? It's something we don't really understand very well yet. However, it's probably worth mentioning that diseases like Crohn's disease or even cardiac failure are associated as conditions to increase collagen signal and deposition in the intestine. So this, we think, could be like a good starting point for interventions, as well as to understand the basic biology of how and why killifish would increase collagen deposition, and how that affects their physiology, and whether reversing it could lead to amelioration. So this is just to show you that killifish could be a good source of information when it comes to age-related changes in functions, molecular patterns, that can be then addressed probably for interventions, and we'll see that in a few slides. One thing that we do in my lab is to try to understand the evolutionary reasons why killifish have evolved this very dramatic short lifespan and this very dramatic and rapid age, collection of age-related transformation over time. And so one of the approaches that we take is the evolutionary approach. We compare killifish species across African killifish phylogeny in particular. Different killifish species have evolved a slightly different life history. Some are long-lived, some are short-lived. So we try to compare and to connect mechanistically even genomics and evolution in these species to their phenotype, and in particular when it comes to age-related changes or lifespan as a phenotype. So we try to connect that with evolution and with ecology. And one insight I want to give you this is work done by a phenomenal former postdoc in the lab, Ray, who now has its own group in China, is a PI in Guangzhou. He's doing great. And so what Ray did was actually to sequence the genome of 46 different killifish species that have slightly different life histories. So some are annuals, some are non-annual, some are short-lived, some are long-lived, some do have diapause, some others don't have diapause. And in particular, this annual life cycle associated with short lifespan is what he was mostly investigating. What Ray discovered And this is why, I'm trying to convince you that even an evolutionary approach is relevant, even for an audience like this one today in Copenhagen, that is interested probably in targets and interventions. Well, an evolutionary approach is useful to us because it tells us not just how certain phenomena like short lifespan or long lifespan can emerge, but also point towards specific genetic targets that have been targeted by natural selection, or maybe have been neglected by natural selection, like in the case of killifish, and that could be used to reverse the aging phenotype. So what we learned from killifish is that small effective population size in an annual species, in species that live in this very harsh environment, is led to accumulation of genome-wide deleterious gene variants, also at genes that matter to the aging process. This phenomenon in particular, and to showcase one such gene is, for example, the case of polymerase gamma. Polymerase gamma is a mitochondrial DNA polymerase, which is nuclearly encoded. And our evolutionary analysis pointed us towards polymerase gamma as a very good candidate gene that had accumulated several deleterious mutations, specifically in short-lived killifish. So what we found during like an heterologous assay that involved the use of yeast as a system was that actually the deleterious gene variants that we predicted to be deleterious in the short-lived killifish were leading to a higher mutation rate in killifish mitochondria. In other words, a killifish is a natural experiment, short-lived killifish are a natural experiment of mutagenesis. And polymerase gamma is one such gene that has been particularly dramatically hit by deleterious gene variants that may lead to age-related changes. We know that mitochondrial replication fidelity and mutation rate in mitochondrial genome, it's very important for the aging process. So in other words, evolution can point us towards candidate genes for interventions for anti-aging studies. Now, I want to slightly shift perspective from evolution to ecology. I have a few minutes left, but our lab is really interested in this. We really are interested in this evolutionary ecological approach to study of aging. So while I hope I convinced you that evolution can be valuable to identify specific genes that have evolved particular variants that may be more or less important for the aging process, and we have many such candidates that we are working on. The ecological process is different. The ecological perspective, ecology has to do with species interactions. And we do think that a perspective in biology of aging that embraces this species-species interaction is particularly fruitful, not just to understand better what it means to age for an organism in the context of interaction with other organisms, but also as an entry point for interventions that can lead to increased resilience and delayed aging, or perhaps even aging reversal. So what is this ecological perspective I'm referring to? Well, like I said, it's a species-species interaction type of perspective. And we hosts, multicellular hosts, interact all the time with unicellular microbial communities that live with us and on which we depend for every single biological process we can think of, from brain functioning to digestion, to defense from predators, to signaling, to co-specifics, to breaking down of nutrients, et cetera, Microbes that dwell on our epithelia are essential to our functioning, our homeostatic processes. So our perspective on this microbe-host interactions into biology of aging is a perspective that asks what is the role of this host microbiota interaction in maintaining homeostasis and in the aging process. And also here, we are very much interested in using killifish because of its experimental accessibility. And the killifish have a very typical microbiota when it comes to microbiome, we can say in this case, when it comes to intestinal vertebrates in particular. And what I mean by a typical microbiome, it's composed mostly of bacteria by 60%, by archaea by about 1%, and by fungi, and viruses by less than 1%. Viruses are largely, bacterial phages, and in particular, if you rank the abundance of the different viruses in the stool microbiome from killifish, you find phages that are the ones that are specific to the most abundant bacterial taxa. Also, we have a lot of unmapped reads you can see here, so we can't quite map all the reads that we get from this metagenomics analysis, which means that there is a lot of work to do still for annotating these genomes better. So this is a typical, freshwater organism microbiome, and the way we're looking into, looking at the microbiome and aging and host interaction is that microbiome, in a way, reflects metabolism. It's a readout of host physiology as well, and the way we look at it is like an extended phenotype of hosts. This is work done by Miriam Popkes and David Willemsen, both PhD students in the lab, and now both turned into postdocs. So they were asking whether the microbiome, this time is not metagenomics, but is 16S or ribosomal RNA sequencing of captive killifish and captive killifish and natural killifish overlaps, and the answer is, to some extent, yes. So there is a vast overlap. There is vast diversity in microbial composition in wild killifish isolates that we collect in the natural environments. And this cloud of dots in this principal coordinate analysis represents the microbial diversity of killifish in captivity, that it's clustered, that they're very similar to one another, unlike the wild captured killifish, but it still, in a way, represents the diversity of the species as a whole, which is kind of interesting and supporting the fact that it's okay to study microbiome of killifish in laboratory. So extending on the concept I earlier raised, that microbes can be seen as a host-specific extended phenotype, if you wish. What we interestingly found is that in natural ponds where killifish live in nature, this is a snapshot, an aerial snapshot from the Gonare Zoo National Park in Zimbabwe, where we do our field work and where killifish live in nature. So what we found is that even in the same ponds, we can find two different killifish species. In this case, the spotted killifish, Notobranchus ortonotus, and the turquoise killifish, Notobranchus furtzeri. Well, their intestinal microbiome is actually different. So it's similar in terms of alpha diversity, in terms of number of microbial species, but the composition is actually different. So that means that indeed microbes in the gut reflect also genetic distance between hosts. This also could be due, of course, to slightly different feeding habits. And so this is something we're very much exploring, dissecting the genetic architecture in the host of microbial composition. Martin, I do have still a few minutes, right? 2 minutes. Okay. Perfect. So we are very much interested in extending our collection of microbial isolates associated with killifish. But what I wanted to tell you is actually the connection between microbiome and health. And work done in the past and spearheaded in the group of Jeff Gordon has shown that lean and obese individuals differ in their microbial community structures, and in particular, healthy individuals have more diverse microbiome compositions. They have more rich microbial communities, and transplanting microbes from lean individuals to germ-free raised mice, as opposed to obese donors, lead actually to dramatic impacts on mouse physiology and health. And so, really harnessing this type of approach, we followed up with studies in killifish, where we first characterize age-dependent changes in the microbiome, where we compare microbiota between young and old killifish. We understand there is a drop in microbial diversity as killifish get older, similar to what we saw before in the Turnbull study by Jeff Gordon's lab. Young versus old killifish have different microbial communities, not just different diversity. This microbial communities enriched in old killifish are enriched for pathogenic proteobacteria, according to annotation. And interestingly, killifish is not just a readout of host physiology and health status, but actually can be used to modulate lifespan and the aging process. And this is actually we tested a few years ago by transplanting microbes from young killifish to middle-aged killifish treated with antibiotics. They received microbes from young donors, same age donors, and just an antibiotic treated group. And we discovered for the first time that we can modulate killifish lifespan by transplanting and by recolonizing hosts microbiota or testing with young microbes. So this led not just to lifespan extension, but also, and I will conclude with that, also to actually maintenance of like higher physical activity measured by spontaneous swimming. There is a reversal to some aspects of innate immune status in the gut, and in particular, anti-inflammatory responses are boosted in killifish that receive young gut microbes. And recent work done by Marcus Böhme in John Cran lab in Cork in Ireland. Like this is probably the most, the biggest group, the biggest, institute working on human microbiome and mouse mirroring microbiome. Actually, they found very similar results in mice as well. So looks like also in mammalian hosts, microbiome changes, microbiome transplantations may be used to revert some aspects of the aging process. With that, I would like to go to the acknowledgement slide. See, it was very ambitious, but before the acknowledgement slide, I would like to tell you that we have 2 junior group leader positions open in Vienna at the Leibniz Institute on Aging. This is actually a research focus on host microbiome interactions, functional host, microbiota host interactions during aging. So if you are approaching the job market as an assistant professor, please contact me. I'd be happy to chat with you and discuss about opportunities. With that, I'd like to thank the whole lab, the funding agencies, Max Planck Institute for Biology of Aging, that hosted my group for all these years, the Leibniz Institute on Aging, who's actually giving us trust and resources. We're very excited for this new adventure. And I'd like to thank you all for your attention. And I hope we have time for a couple of questions.
Thank you so much. Thank you. Really great talk, Dario. Maybe we can start with a question from the online audience. Vardan Saroyan is asking, can aging in killifish be an evolutionary adaptive mechanism of fitness to permanently changing conditions?
Yeah, thank you very much for your question. So permanently changing conditions would require that would not necessarily require that organisms would live short, right? So changing conditions may require that organisms are able to reach sexual maturation very fast. And this is exactly what killifish do. In fact, they seem to have maximized timing to sexual maturation. And irrespective of how long they live, timing of sexual maturation is the same across killifish. So there is no trade-off between how fast you develop and how fast you age in killifish. This is a very important thing to understand about this species. The ones that live shorter simply start dying off earlier and start displaying like a larger set of age-related transformation earlier than the others. I hope that answered the question.
Very interesting. We have a question from Felipe here in the audience.
Hi, Felipe.
Thank you for your talk. It was a very interesting talk. I was very curious about the changes in collagen and hydroxyproline. So do you know whether there's a role for prolyl hydroxylases in HIF in your phenotype? And the second question to that would be, do you think oxygen tension is playing a role? And if there is, do you think is the microbiome that somehow, by changing with age, is actually triggering the collagen effect?
Yeah, Felipe, we really don't know what's happening. So we do see this, and this is a very striking phenomenon. We do see it over and over. I mean, we already found it a few years ago with Jens's work, and Miriam found it again now. And this is very striking accumulation. Of course, the question is, why is collagen accumulating, right? So you have constant production and less of, lysis of collagen, so you're less able to remodel the extracellular matrix. So then we're looking at metalloproteases, but the role of the microbiota in this also we don't quite understand. We're actually trying to see whether microbiota transfer do reverse the collagen phenotype. We don't know. We don't have an answer to this yet. Yeah, whether the microbiome plays a role, like I said, we don't know. Collagen deposition, also fibrosis, is a typical phenotype associated also with inflammation. So it's kind of like, could be that systemic inflammation may be associated with this collagen scars and fibrotic phenotypes, but the etiology of this it's still to be clearly understood. Yeah.
Great.
Yeah, I could answer your question. Yeah.
Thank you, Dario. Let's give Dario another round of applause. Wonderful talk.